we're so happy you're here for the first one. A couple announcements before we begin. I'm Grace. I work in the whale lab, the lab with the whale on it and the touch tank. You probably walked past 100 times. You're always welcome to come in and say hi. I work primarily with the field trips department, so doing our educational program development, doing things like conferences and workshops, and I get to do like all of our fun events like summer seminar series. So you may see me in and around town, about town. This week is also Ocean Week. It's Ocean Week here in Canada from the 1st through the 8th. At BMSC, we're hosting 3 events. The first one is right here, right now. You've made it. Congratulations. Tomorrow, we are hosting a beach cleanup at Pachena Bay. We'll plan for folks who are interested to meet us there.
Sorry, keep going. Just hold it if you want.
Oh, my bad. We're hosting a beach cleanup tomorrow, Wednesday at 5 p.m. at Pachena Bay, just 10 minutes down the road. We'll be parking at the campground side, so the Pachena Bay campground side rather than the national park side, but we'll see on the beach. Kirsten and I will both be there, and we'll be in our high-vis bags and gloves provided, and it'll just be a gorgeous evening on the beach, taking care of the places that we spend all our time. Then Thursday is back here, 7 p.m., same time, same place, but different speaker. I'm actually our speaker for Thursday. I created like a very silly, fun, interactive talk on the deep sea, and we'll point and stare at all these amazing deep sea creatures and talk about how we protect it and just all the crazy stuff going on down there. So you are so welcome and would be greatly appreciated your attendance. You're so invited. Okay, our speaker tonight is Dr. John Reynolds. Many of you are very familiar, so I'll actually just invite him up. But we are so grateful for you sharing your talk this evening. Thank you so much for being here, and thank y'all for attending. Please welcome our speaker.
Thank you. And yeah, we appreciate it. It's a nice sunny evening. Game 1 of the Stanley Cup. This is forget it. No looking at your phones.
It's tied 3-3.
Okay. It's particularly impressive score here. So yeah, I'm happy to talk about some of the work. Well, given that it's a game, so I'm going to talk about our work on we've been doing for a long time up in the Great Barrier Rainforest. My position is technically it's a leadership chair in salmon conservation, and so when I was first setting this up, trying to figure out what it would look like, I got very interested in the story of how nutrients from salmon might be supporting other components of the ecosystems. But anyway, I'm very keen to give a talk about that. And also, by the way, if you ever want to find me, I'm just here on the top of the Ms. leaderboard. Just in case you're wondering, I expect to be displaced after I leave on Friday. Right, so with a lot of people tend to specialize on one type of habitat, and many people see these different habitats differently. So here we have an estuary, out here we've got the ocean, of course, and people would look at this in very different ways. And terrestrial ecologists, for example, like I said, put them in the water if they can avoid it. They have their own journals, their own meetings, their own conferences, their colleagues, and so on. So they would stick to the back end there. Freshwater people would do the sort of things that we've been doing in our course, where we're constantly measuring water flow, stream profiles, all the sorts of things that we've been doing in this course are standard things, and then people can apply that to understanding the distributions of fish or stream invertebrates or what have you. And of course, marine people would be out roughly where you are looking at it. Of course, they're looking at nutrient changes in the marine environment, especially if it's an estuary. And the fun thing about salmon is that they knit all 3 of these habitats together. So if you're going to study salmon, you could just study them in the ocean, and some people do. And you could just study them in the fresh water and count the fish every fall, as many people do. But if your question is, what is their role in the wider ecosystem, then you actually have to cover all 3 habitats. And not many people actually do that as a sort of generalist naturalist. This suited me quite well. And so I set up a program when I just decided to take up this chair at SFU, looking at that. And this is the kind of salmon I study. I study dead salmon, and as people will know, if you're with me when we're looking at fish on stream, I had a pretty hard time identifying a lot of the juvenile salmon. I don't really work on them much. I'm I'm pretty good at identifying dead salmon, though, because that's the main one that we work on. So I'm interested in how salmon knit these different habitats together, because when they are killed or when they die, their carcasses often end up in the forest. And just a reminder of the distribution of salmon, they go all the way around on the North Pacific. We have most of the same species on both sides. So you could go to Kamchatka Peninsula, for example, and see spawning sockeye and steelhead and all the sorts of fish that we're very interested on this side of the Pacific. This is a photo that you can see in lots of places online, and in fact, this happens at a hatchery near my home in Vancouver area. This person is wearing gloves. That's your first clue to what's going on. This is a frozen salmon that he's about to throw into the river because this has come from a hatchery. So we have a whole truckload of dead salmon. And these fish were all, unfortunately for them, they were the source of the next brood stock of the salmon hatchery. So people catch the fish, they kill them, they strip out their gametes, mix them together, and breed them. And so what he's doing is throwing the carcass back into the river, because the understanding, the theory is that the carcasses put nutrients into the rivers, which can then work their way up through the food web and maybe ultimately support the next generation of salmon. It is an interesting concept because many of us from urban areas are used to problems with eutrophication in the rivers. Too much nutrients, too much phosphorus in particular, right? This is the big problem in a lot of freshwater water bodies. But in fact, in the Pacific Northwest, as long as you're away from a lot of people, in fact, it's the other way around. They can often be quite nutrient poor. The natural state is to have very little nutrients. And so it's possible that these carcasses, when they rot, they'll be feeding the vertebrates, which could then feed the next generation of salmon, and so on. But this is another look at, there's another side to salmon, and actually you might even be able to hear that. This is in one of our study rivers in Belle. I'll introduce the territory to you in a moment. Here you have two male chum salmon fighting for position. That's probably a female up here. So the males are trying to be first to spawn when she's ready to go. So she's digging a nest, and you can see it's full of salmon that are all fighting, and they're stirring up the gravel. And it's actually quite striking. You can go into one of these things and you can see the areas where the fish are spawning and where they're not, because the rocks are green where they're not spawning, and they've been scraped clean by their digging action. And so there's actually been studies showing that you can get so many invertebrates that'll wash out of the system because of that. They get suspended and leave. In fact, maybe there's a net nutrient loss due to salmon. They bring nutrients back in with their bodies when they feed at sea, because most of their weight is put on at sea. But then you have this effect. And I remember getting corrected on this point once. I met with some people at the University of Washington, told them what I was studying, and immediately when I said I was studying the fertilization effect, immediately the person cut me off and said, you mean the perturbation effect, you mean the stream engineering? Because in his opinion, it was the other way, that the salmon could be reducing nutrients in streams. And I was lucky. I was previously in England for a number of years, a professor at the University of East Anglia. And when I decided to move to the West Coast to take up this position at SRU, I told some people that I was planning on doing this, and people from the Department of Fisheries and Oceans said, well, good timing, because we've got this policy that's about to come out. They had worked on it for about 10 years. And the policy sets out the blueprint for what Canada's trying to do with West Coast salmon. What are they for? How are they going to study them? What are their conservation priorities? And so on. And as a result of a huge amount of input from environmental groups, they had a strategy which actually looked at ecosystem connections with salmon. So the challenge, as they said, is the need for development of an ecosystem objective that's widely appreciated but difficult to quantify. That's a fair statement. Difficult to quantify. Yes, they get that salmon are there for more than just fishermen, fisherwomen, but they're not, many things eat them, but it's difficult to quantify that. And so they said few studies provide advice on the number of salmon necessary for a healthy ecosystem. And they were very smart because they didn't define what a healthy ecosystem means. You can imagine that could have delayed it another 5 years to try to get scientists to agree what the So I was very fortunate to be allowed to work in Celtic traditional territory in the Great Bear Rainforest. You can see the so-called Great Bear Rainforest stretches here. It's pretty green compared to a lot of the rest of BC because this has been logged very hard, as we know around here. But this is an area that's hard to get to. There's been very little industrial logging. Yeah, there is some for sure. You can certainly there is some brown areas here that people can get in from waterways. There's been historical logging, but not large scale logging. Definitely in very much of the Celtic territory, nor of the other three nations in the Central Coast. So I was really fortunate. I got in touch with some people in the band in Bella Bella, and I was aided very much by Morgan Hawking, who was my first postdoc, who had worked there. He introduced me to some people. They took us out for a day or two in boats, and we looked around, and one thing led to another, and I've ended up working with data from all of these sites, all these locations. So each dot is a stream, and there's my scale 20K. So the village of Bella Bella is here. That's the main base, is about 900 to 1,100 people living here. And then one island over, we have Shearwater, which is it's a marina. It's a place where people can stop their boat, can stop in when they're going up further up toward Alaska. So we work in most of these streams. The color codes are just from at that time who was responsible for counting fish. So different colors mean different things, but we have certainly done work in all of these streams. And the farthest we go for a stream count or stream work would be maybe an hour and a half by boat. That's how far we go. And this is one of the boats that I've been using, one of my boats, research boats. Howard Humjet here has led our team for about 10 years. He's from the village, and he's kept us safe. He's trained successive graduate students to take over the driving and captaining of the boat, and this has been extremely important. He's also been our head fish counter. So when we count the fish, the person who goes first has a certain duty to certain fish they're counting. He's the lead, and then they'll have two people at his flanks. And so he directs our traffic, and he's moved on to a different job now, but we work very closely with other people from the nation, and all of the data that we collect are done in collaboration with the nation. They have copies of all of our data, all of our physical data on the streams, as well as of course, the salmon data. And we work with DFO figuring out who's going to capture streams too. We give them the worst streams. They do the really big ones where we just don't even want to go near them. We tend to do smaller ones. This is a picture of Roscoe Inlet, a beautiful inland fjord. And the streams that we chose really were quite variable. This was from Roscoe Inlet. This is fairly wide by my standards, kind of similar in some ways to the streams that we've been looking at in this class, maybe the Nitnat. And so they can be fairly wide, or they can be very narrow. Sometimes some of them are only 2 meters wide. And we're primarily looking at chum and pink salmon. That's they're the dominant fish in these systems. So that's largely what we're studying. And we're interested in 3 large carnivores. The Great Bear Rainforest has arguably the tag large carnivore assemblage in the world. So even we think the Arctic is remote or not very impacted, but even compared to the Arctic, here basically most of the First Nations are not interested in hunting. In fact, many are dead set against commercial trophy hunting, which used to happen until many of the licenses got bought up by NGOs in partnership with the nations. And so basically nobody's there now in very large tracts of the Great Bear Rainforest. Nobody's shooting these things. And so we have wolves, which are marine animals on the coast. They are eating a lot of marine fish. They actually grab the fish, pull them right out of the streams. And we have both bears. The grizzly bears are more on the mainland sites. Black bears are more on the island sites. And this is a very typical thing that you can see at an estuary. So here's the chum salmon, and you can see my book for scale, and you can see they've left behind an awful lot of meat here. And this is, to me, that I'm seeing a huge sack of nutrients. So wolves can just leave hundreds, literally hundreds overnight. You come back and some of the fish are still, they clearly were killed overnight. They can actually kill hundreds of fish in the estuary and eat very little of them. And I want to play this video. We count a fish, as I mentioned. Here's how we do it. Let's see. So some people are counting dead fish. We record what happened to it, whether it died of natural causes or was killed by a predator. In the lead there, that was probably Howard. In fact, up in the lead, you probably may have heard somebody yelling to warn the bears that were coming. That's kind of our standard, here we come, yelling to the bear. Last fall, we got chased out of a stream twice by a bear. It just would not let us do it. And so there were 5 of us. And we still said, no, this isn't worth it. Backed out, and it was an hour boat drive to get there. We came back 3 days later, and the bear was still there, and it still wouldn't let us do it. And we left again. So on the third, by then I had finished and gone back to Vancouver. On the third day, the team was able to get into the street. I know, it was really bizarre. It wasn't even a very big bear either. It was a black bear. So we have looked at many routes through the freshwater system, from biofilm and algae. We looked at the effects that the fish are having on these. We can also look at how they're nutrified by looking at N15, the stable isotope of nitrogen that is amplified by fish. Lots and lots of stream invertebrate work, and we've looked at effects on juvenile coho. I'll mention that in a moment, and 2 species of sculpins as well. And then we've also done our terrestrial route here. We've done some work on bears, a lot of work on birds. I'll mention that, and plants as well. And we've also, there's another axis I'm not going to bother trying to fit it in. We've also done, I've imagined an axis here for the marine pathways. We've done a lot of work in estuaries, tracing the nutrients from everything from clams, mussels, and even Dungeness crabs. So I'm going to give 4 examples of impacts that we were testing. And the first one was that fish one. Remember the kid that was throwing the frozen carcass? Well, that story just seemed too good to be true. The idea that you can sort of jumpstart a stream by putting carcasses in it. And for us, in fact, it probably could not work for most of the species, because pink and chum salmon do not feed when they're juveniles in the river. As soon as they resorb their yolk sac in the spring, they leave. So they're not hanging around. They cannot be feeding on anything that was fertilized by the generation that went before it. But coho do stay. And so here's a coho. We've seen lots of those in this course, and as you know, we've only so far, I think, seen 1 age class, which would be the ones that were born this winter, that came out of the gravel early this spring. But they can even stay for 2 years. And they're really distinct size differences if they're in their 2nd year. So we did some work on those, and this is work by Michelle Nelson for PhD. And here you have the number of pink salmon that were spawning in each stream. And here you have the abundance of coho on an index. And so basically you can count these coho, and our class certainly saw that. In decent sized pools, and these are really small streams, you can actually quantify this. And so you can see that when you have a large number of pink salmon, the coho tend to be more abundant in these streams. It's quite variable. If there are no fish, sometimes there are no salmon because there are no pink or chum salmon because they're not as good jumpers as the adults, as an adult coho. Coho adults are the sort of most athletic of the fish. So we can get to places where there will be coho salmon, but there's been no impact of the nutrients from pink and chum. And we, to Michelle's credit, we looked at 17 habitat variables that might have confounded these analyses from our stream measurements. And no matter how we looked at it, pink, salmon, and secondarily chum abundance were the best correlates that we had of coho. I can't swear that there isn't some common other variables. Maybe the stream has to be semi-decent for salmon at all for them to even be there, but it certainly won the competition. And so then we moved on to birds. And this took a little bit of time. My initial idea was to count birds that we would have and using standard censuses on the streams. But that was a little tricky. We couldn't hear over the sounds of streams. We did eventually solve that problem. But before we did, Rachel Field joined me for a master's. And so she had a background in forest plots, forest analyses, as well as being a good birder. And so we looked at the forest birds around estuaries. So we didn't have to deal with our running water problem. And so we would just anchor the boat, and then Rachel did this work on quantifying the vegetation, all the confounding variables, recording the birds in a very systematic way. She also did the ones that were feeding in the estuaries, and so there's a not too surprising result here, so that the estuaries in the streams that have the most biomass of carcasses also have the most birds that are feeding in there, various species of gulls, eagles, ravens, and so on. So that makes sense. And by the way, these are not all separate streams. She did multiple visits to each stream. So that's not too surprising. You don't even have to get out of the boat to do this one. You can just, in fact, she didn't, so she wouldn't scare me, just go up quietly, survey the total number of birds. So that's not a shocking one. We were pleased or interested to see, though, that the one thing we weren't expecting is that eagles were especially associated with fresh carcasses, not dead. Not just any old senescent carcass, which can be pretty disgusting. I don't blame them. They go for the fresh carcasses. Gulls and so on are happy with the other ones, but fresh-killed, bear-killed carcasses were the most correlated with the eagle abundance. But this is the one I really liked, where we quantified the birds in the breeding season. And so again, it's almost all by ear. And here you see the pink and chum combined biomass. And this would be one stream. This is our biggest stream, the one with the most fish. And 2 visits in 2 different years. Each 1 year, second year, 1 year, another year. So you can see there's pairs of data points. If you have very few fish or none, because it's a waterfall blocking, then extremely variable results. Otherwise, in the big ones, we got no birds. So first time it's shown that you can get a higher density of breeding birds around estuaries that have more fish. But the thing that was interesting to me especially is that some of these birds are migratory, like Townsend's warbler, western flycatcher. They don't feed, they're not even there with the salmon. So they're not feeding on the carcasses. They're not feeding on flies that are feeding on the carcasses. These birds are probably benefiting, we think, just through overall enhanced insect abundance in the streams and with more fish. And it does not have anything to do with forest structure. You could rule that out based on our forest plots, because the forest structure has a huge effect on the density of birds. We didn't get that in the study. So this is, well, I'll keep the story short, but Kirsten Wilcox was a field assistant to another PhD student studying birds, whose work I'm not going to bother mentioning right here. Kirsten was studying the hardest bird in the world to study, which is the Pacific wren. That's the one that we have around here. They're hard because they are in deep forest. Their nests are very hard to find. They nest in moss balls sometimes, or under root balls, and the males make dummy nests, so you can spend all day following one of these birds. You think, finally, I got the nest, and then nothing ever happens, because it was a courtship nest or something like that. They may end up not even happening. So anyway, she did it really well and had an army of field assistants tracking these birds. And I'm going to mention how we did the stream mapping. She color banded as many birds as she could, but the real question was whether we could look at the territoriality of these birds. So here's how the territory mapping works, and this is her artistry here with banded birds yelling at each other. They don't really do that. Here's how it works. So these are actual data here. If you thought your stream mapping data were pretty in this course, there you go. How's that? So Kirsten would have walked into the stream. This is a schematic of where the stream was. And this is a place where she has encountered a Pacific wren. It's probably either yelling at her or yelling at another bird or singing, but they have a call note that's very distinctive. And she also had one here, and encountered here, and here. And then some distance away, there was this one over here. She knows they were different birds because she could hear them at the same time. And that was basically all she got from that first visit. This is one morning of work. So then if you go back, again, we're not looking at banded birds. You go back and here's her here's her data about 3 days later. Again, there's some movement, moving around a bit. There's one at this end, there's one bird up at this end, and nobody in between. And so with successive visits, you can build up the map of their territories. So pretty clearly, there's two birds along this stream. One that had huge territories. One goes from here to here, and one sort of wraps around from here up to here. And they occasionally do probably invade each other's territories, but that one was pretty discreet. And this particular place was a spot where there were no salmon at all. So there was a waterfall, no salmon, and here is how we're able to draw the two territories. There is a little no man's land, no wren's land in between the two territories. And then here an example from some of the other streams. She had quite a few streams. You can see the one that has a really large number of fish has a lot more territories. They're much smaller. They have to be if you're going to pack them in. Interestingly, there's something you can sort of see here. It held up statistically. They were actually more selective of where they put their territories. So some of these big gaps are actually real. They are real, and they were more selective in the kinds of trees that they were wrapping their territories around, or the densities of trees for their territories. These are obviously right next to each other and overlapping, but in many cases they were more selective when there were more birds. And the males are, extremely busy in these ones here. If there's only two birds, they don't have to talk very often to remind each other that they're still there. These things, it's just mayhem. They are so busy fighting to maintain these territories. And she used a camera on the end of a tube to go into the nests, the ones she could reach, or into the stumps, and she was able to actually look at the number of young in many cases. And after they fledge, they stay with the mom for a little while, and she could also therefore figure out how many were coming up per nest. And the number that were coming up per nest were about the same, whether they were in the high density salmon stream or the low density stream. That means overall, this Claxi here, this stream, is producing a heck of a lot more wrens than that one. But the individual reproductive success was equal, whichever one they were in. So the wrens were distributing themselves among the resources in a way that ended up equalizing their fitness. Okay, so plants. Again, I hope our Banfield class isn't tired of hearing about plants. Everybody know what that is? Well, you know it's a blueberry. It's one of the blueberries, obviously. So that's the oval leaf one with the red, where the red stems. I didn't see that nearly as often as the other one. But I was very interested in these. There are no plants, there are no emergent plants in the streams. They would be washed out. So I was interested in the plants on the riparian zone. And this was when we began this whole thing with a study of a stream called Ripley. I actually showed Ripley earlier. That's the one that has no fish in it because of this waterfall, which is right near the mouth of the river. And so salmon can't get up this, even coho can't manage that one. And so there's no fish at all. And so this was when my postdoc Morgan Hawking and I got the bright idea that we would go and put fish at the top of this waterfall. So we went by boat, went to a more productive stream, grabbed these carcasses, put them in plastic bags, and walked up this bushwhacked up over the waterfall through the forest, and we planted them out in the middle of grizzly bear country. And so here's what it looks like. So we would drop a carcass and flag it, and then we would take a sample of plants, either right beside the carcass or a meter or so away from the carcass. So that was our before. We wanted to look at the nitrogen content and things like that in the plants. So this is the start of the experiment. You can see how disgusting these carcasses are that we were putting out there. And then we would come back again in the fall when there would be a skeleton. In the spring, sorry, this is the fall. In the spring, we come back, it's just a skeleton, we do it again. And here is one of the results of this experiment. It's the first time anybody ever done this kind of experiment. It may be because it's so disgusting that nobody else in their right mind would want to do this. But here you see the percent nitrogen in 3 examples of plants. And so there's the percent nitrogen, and here's the fall at the beginning when we did the experiment. No difference in percent nitrogen in leaves, of course, because there hasn't been an effect yet. This is the day we put down the carcass. In the spring, there's a jump beside the carcass, much less of a jump away from the carcass, so there is a bit of an increase too. So you have this bounce that happens in the that we can detect in the spring, even though we put the carcass down in the fall when the plants aren't really growing very much. There's obviously some sort of effect through the soil, and the plants are taking it up and having a higher percent nitrogen in the carcasses as a result. So that's the first time anybody has shown experimentally what a lot of people thought probably happens, but we didn't, there was no evidence of it. And the one thing I really liked was that we did this at a lot of different sites. And here you see 2 things here, 2 species, of moss and stream salmon density. So these are the number of fish that are naturally spawning in the streams. And so if you have no fish at all, then you get the lines are fairly far apart. The experiment has a bigger effect, like it did at that waterfall site. But then in streams that have regularly get a lot of fish, we're having less of an effect from our manipulation, which is what you'd expect because there's already probably a good nitrogen base in the soil. And so this fertilizing effect is less important. But that's percent N. So this is where I got really interested. We never wanted to stop there. The real prize, I thought, was to see if you could see a change in species composition. And so this is where the great Quadrat Fest happened. And again, I'm sorry if this brings PTSD to everybody in the class, but we put down these quadrats perpendicular to the stream. Did a lot of these. We went to 50 separate streams to do the work. There's Morgan Hawking, who led this study, my postdoc. And here's the transect tape, and we looked at the diameter crest height of over, I think it was 6,000 trees. We had over 30,000 lines of data from our quadrats. You can generate a lot of data this way if you go to 50 different watersheds. And so we were able to predict which ones would respond, which ones would be more common in streams that had more salmon, because they were already known from other sources. We knew that some of these things like false lily of the valley, salmonberry, and stink currant were all in typical of nutrient-rich soils. And in fact, I think these 3 were very dominant in the 2 in that and beside it. The Cereda, again, very rich floodplains there. And then Salal, false azalea, red huckleberry, they can grow just about anywhere. They're not dependent on that kind of external source of nitrogen. So we predicted there'd be a shift in the community composition from as the number of salmon increased across rivers toward the nitrogen-rich ones. But we also looked at the N15, which is the nitrogen isotope that is amplified when it's been coming through a marine source, such as salmon, especially a high trophic level source. And so here are examples from 2 nutrient-poor indicators. So these are 2 species where, in fact, they are taking up salmon-derived nutrients. So that's what we're seeing here. Each dot is a different stream. And so we had 50 streams, and you can see from 0 to 30 kilograms per meter squared of fish, they're taking it up. So these nitrogen poor indicators are taking it up. And then so is everybody. False lily, there's salmonberry. Very similar, strikingly similar curves, kind of a surprise. But then if you look at the percent nitrogen, so do they have more nitrogen? They may have more of the isotope, but is it just kicking out the other isotope? And the answer is there's no pattern whatsoever for the nutrient poor indicators. These are not, they may be taking it up, but it's not increasing the nitrogen in the leaves, whereas these 2, it is increasing it, and salmonberry, at least at the highest densities, showed reasonable response. And then if you look at the densities, here's this community shift that we were predicting we would find. So we have a drop in the
they're called ericaceous, members of the family Ericaceae, like azalea and blueberry. They're dropping, no effect on false lily, and the salmonberry are increasing. Lots of scatter, and I'm going to talk about that in a minute, but salmonberry densities are higher. We've subsequently done a study where we actually found that salmonberries have more berries per plant when there's more salmon in the streams, and but the sugar content does not differ. And so we, because they're in the same genus as raspberries, we were able to, there was tests available for how to look at the sugar content, how to process them. You squeeze the juice out of them. We went through the whole rigmarole and found no difference in sugar content, but we did find a difference in the berries per plant. And so here's a way to put this all together. Here's, if we array all of the plants on an axis that describes their affinities for being a nutrient-rich indicator, then you go from a very nutrient-rich community to a very nutrient-rich poor community. So it's not just these two plants, it's putting it all together and then relating that to the density of the stream salmon. And you can see that, again, lots of scatter at the low line. So there's other things controlling these things at the low numbers. And then it starts to rise pretty nicely at the top. When you're doing this, it's a correlative study. So of course we're thinking, yeah, well, okay, you know, it's not experimental. I showed you an experimental one. And I will never forget, actually, when Morgan came to my office at SFU, and he had just done an analysis because we were there to just see if we could understand the variation, you know, above and below the line. And it turned out that two key things seemed to explain most of that variation, or a lot of it. One, and they're both to do with other things in the streams. One was that these really steep gradient streams don't tend to benefit from salmon nutrients very much. So the nutrients are not going to be retained on a bank like this. And we found that these sorts of streams had really very little effect on the salmon. Didn't matter if they had a lot of salmon or a few. If it was like one of these canyon type streams, we saw no real effect on the really steep banks. And then the other one, thank goodness we thought of this beforehand, was that the density of red alder was important because that's a nitrogen fixer. This will actually produces its own nitrogen, makes it available, bioavailable in the soil. And so we had measured red alder in case that was the case. And sure enough, if you have a lot of red alder, it doesn't matter how many salmon you have in the stream, you're not going to see an increase in nutrient-rich indicator species because they're already there because they've already got a different source of salmon. So it was the first, I mean, I don't want to overstate this story. I think if you took me to a salmon stream, I could probably tell you if they have very few or middle amount or a large amount. That's about as far as I would go. I think I could, at least in that region, I think I could do it. But we have to, I don't want you to think, as some people do, that if the salmon disappear, the whole thing's going to collapse and that everything's going to change and everything. It's a small effect, and it probably doesn't extend much more than about 50 meters from the stream. So there's a lot of wear and tear on the researchers when you're boating to multiple sites per day, different site, all the time. And I had this fantasy that it would be really nice if we could just stay in one place and do something in one place only. And the thing I really had always wanted to do was to work in salt marshes because they're just beautiful, lots of plants, and if we could be in one place, we could get a lot more done and save some fuel and everything else. And along came Alison Dennard to do her PhD with me, and let's fly over Goat Bushu salt marsh. Here, this is her study area. See the dots? You see these arrays here? I'll tell you about those in a minute. This is Goat Bushu. It's about a half hour from where we are. So you anchor the boat out here, and then you hike up, and then you get to these places where we have these patterns. And this is because we designed an experiment where we did 4 different treatments in a block like that. So there was a trail leading from one block to the next. That's what that trail was. And then there were blocks. Those dots were the blocks where the actual experiments were going. This was one of the riskiest things ever. We had a plan B. reason it didn't work, because we didn't know, first of all, for sure that if we put out carcasses in these blocks, that they would stay there. Partly because it's a saltmarsh, so you can get a really high tide, so we staked them down and netted them and hoped the bears would leave them alone. And it was also risky because of this flooding thing, it could be that would swamp the plots, and therefore any carcass you put down might have no effect. So we had a plan B and a plan C, but we just had to try it. And so here's Allison. You see there's a theme here, us and rotten fish. So this was an all hands on deck thing. She got everybody to join with her. They brought these carcasses here because there weren't very many in the stream. They didn't want to have a high background level. And then she had 25 plots. There's meter by meter plot. Each square was a meter. And so there were 4 of these 1 meter squares things. And so we had a control where she did nothing, added a salmon, added fucus, rockweed, or added both salmon and rockweed. So that's the plot, that's the treatment. 25 replicates of this thing, and she did it for 3 years. And I brought in a co-supervisor who Elizabeth Nelly, who knew a lot about the pollinators, could identify bees and whatnot, because we wanted to see if it affected the plants, but also if it affected the pollinators of the plants. So we chose 4 focal species for most of the work, and these were all in full bloom in September and October when the work was being done. And so we focused on these 4 things in her plots, and then Allison and an assistant would count all the visitors that came to the flowers. It was mostly flies and bumblebees. And so literally you would stand there on a sunny day, usually, because that's when you see bees, and she would record all the visits that were happening, and first in one plot, then she'd go to a new one, and so on. And so it gets, although it's a beautifully elegant design that she came up with, it gets a little tricky to disentangle what is affecting what. I'm going to make a long story very short, and this work has been published. Most of the plants had larger flowers in the plots that received the salmon, whether the salmon was with fucus or not. Bigger flowers for the most part. Many of them, some of them had higher seed sac as well later on. And then as far as the floral visits, this is just one path that was shown. So here, yarrow had 65% more stems in the plot where she dropped the carcass, and 6% more visits, floral visits, by insects. So that treatment had this pathway of what we believe is causation. I think it has to be because it was an experiment, whether we had control or what we had put down. And so each of these treatments has arrows. This has many arrows, and the whole thing could be a graph full of arrows, but it has been published. But we have published it. If you want the details, you can get it there. But it's the first time someone has shown that salmon can have an ecosystem effect that can go up, not only through plants, very few experimental studies of plants, but then also have this secondary effect on visitors. So I want to just come back to the context of my study here. I have not shown you a lot of the things we've done. I can assert some of the things here about check mark meaning yes, effects were as predicted for sculpins and things like that. And I talked about the birds. We've done a lot of work on grizzly bears with Kyle Vitale, who did a PhD, co-supervised by me and by Chris Darramont. And so I want to go back to how one of the things that coincidentally was inspiring me, I thought this was just going to be a fun ecology study, and then same as I was trying to put it together, the Department of Fisheries and Oceans had come up with this goal of trying to develop an ecosystem objective. We have done a lot of work on this, in which we've now summarized studies by other people. And so I'm not going to show you the graph, but we wrote a big review of this, where we found a very large number of published graphs, ours and other people's, looking at different aspects of this. We redid all the analyses from all of the graphs. This is led by Jessica Walsh, who is now a professor at the University of Queensland. And we looked looked at how many times, basically what is, well, we're trying to answer this question, where is it? How many salmon do you need? And so what we asked was, what does it look like? How often do you get an asymptote like we do? Answer is not very often. Most studies don't have enough fish, I think, to have found that asymptote. The people working on bears from Alaska have certainly found them. And it turns out that there's no magic number. For a while, I thought we had a magic number just based on our own work. I could say fish per meter squared or that sort of thing. But no, as you can imagine, college is not that simple. It depends on what you care about. If you care about birds, it's one number. If you care about coho salmon, growth rate, density, it's another number, and so on. But we've got those numbers. Those numbers are now in the literature, and they're now summarized. In the meantime, that poor old wild salmon policy never got implemented. So people just decided it was nobody's job to implement it. And in fact, there was a federal judicial inquiry into the loss of salmon, sockeye salmon from the Fraser River, where the Commissioner Cohen repeatedly chastised the Department of Fisheries and Oceans for not implementing their own policy. He pointed out nobody was responsible. It was nobody's job to implement it. And so it kind of languished. We attacked this from many directions. Jessica Walsh is the lead author of this. She's the one who did this big review. We've tried many ways. I've talked to various people about this. I've various ministers of fisheries and so on. And the bottom line is that people do appreciate that salmon are important for more than just people, but things have shifted. The number of salmon that are coming back in southern rivers has declined. It's become less. I guess it would be a luxury to be reducing fisheries in order to help bears or eagles. At this point, it's all hands on deck for a lot of southern salmon streams, in the Fraser in particular, and a lot of populations, some of which are endangered. They're actually endangered with extinction. So the ground has shifted in the 20 years since that policy came out, and finding enough salmon for healthy ecosystems would be great, but right now, a lot of people are just trying to find enough salmon to have the next generation of salmon survive, and fisheries, unfortunately, for the people who depend on them, are very often not even open. We don't have the openings that we used to for fisheries. But there are still some great runs, there's still very healthy populations, and definitely I think what I've learned is that ecosystem study subsidies can translate into management decisions. But people do have to decide what the targets and trade-offs are, and how much they're willing to give up in order for it to happen. So I'll leave it there with a shout out to an army of students and postdocs who have supported this work over the years. That's only from the first 3 years of our work. So it's gone on ever since, and is continuing in partnership with them. So I'll leave it there. Should we have a hockey update?
I was looking at my phone.
Oh, yeah.
I'll take a look right now.
Okay. Anyway, I'm happy to answer any questions if people have them. Oh.
It's 5-4 for the Knights, 2.5 minutes left.
Okay. All right. I think we know who you're voting for. Yeah. Any other questions? Yes.
Do we know if the insect population, or sorry, if the community changed as well, or did they just look at like the number of visits?
We just looked at the number of visits within, an experimental array. So we didn't have like a stream-wide comparison.
Okay.
So that we don't know. But I'll tell you something we do know that I haven't mentioned. We did a study of parasitoid wasps. I never thought my ecological journey would lead me here. Parasite wasps that parasitize the maggots of the flies that eat the carcasses, right? So if you see, often you'll see a carcass and it's just writhing with maggots. Well, there's a parasitic wasp. So these wasps are more common. We did traps for them. They're more common on streams that have more fish. It's not too surprising because they're eating these sort of maggots. But we also did the N15, the stable isotope trophic signature. And so they are above killer whales. Because if you think about it, southern resident killer whales eat salmon, just like flies eat salmon. So they're equal at that rate. But they're one above that because they eat flies that eat the salmon. So it's as if killer whale had a predator, its isotopic signature would be the same as this little tiny parasitic mollusc. So we still haven't found a way to capture that excitement in a scientific paper, but it is like
Very cool.
Yeah.
Parasitoid wasps are very cool, so.
right.
Yeah.
Okay.
Thank you.
Yes.
Do you know why wolves are not eating the whole salmon? Like, I've never seen that, just the head chopped off. Maybe that's
Yeah.
not always, like, it's not always that they go for the head, but I don't know, I don't have any background in like wolf or salmon ecology, like
They
why?
usually only go for the head. They do that crunching up the head, and sometimes it's just the brain that they're eating. And the theory is that there may be a parasite in the musculature that the wolves are selected to avoid. So there may be something parasitic, but I've never been able to track that down, and I don't know if that's truly it or not. But that's why we can tell who killed it. Most of the time, you can tell who killed the fish, because if it's a bear, they will take the musculature. They won't eat all of it, but they're quite a different foraging pattern from wolves. Yeah. I should find that out, because I get asked this every time I show that photo, and I've just gotten so used to the fact that, oh, there's a wolf kill. But I've never figured it out.
My other question is, when you were surveying the birds near the waterfall, how did you overcome the noise of the waterfall?
Yeah, so at first we avoided even doing stream work with birds because of that problem. We don't work right beside a waterfall. 5 of our streams have a waterfall, and we avoided being near the waterfall. But after a while, I sort of realized that these rainforests are just dripping with moss, and they're kind of like a soundproof room. They really actually, you notice when you're talking to somebody in the forest how different it is. And so if we just we did point counts, so we had randomized selection of point counts, and we would have to go find that random location. And so we didn't have them right beside the stream. Sometimes it would be we could survive, but usually they weren't right on the stream. So there's a whole other study that I haven't actually talked about, where we actually looked at the densities of the forest birds, the birds right in the forest. We had similar results, but we didn't have to. The only thing that was hard there was getting to those point count locations. It was like looking for a geocache, your GPS, and when we chose them randomly, there was no consideration of whether there might be a tree that's this high lying down in your way, and yeah, they were not a lot of fun.
Just one second. So for the study where there's 4 plots in the transect, I feel like isn't the plots really close by, so they affect each other, like how the salmon's the nutrient will be covered?
That's a great question. There could be some spillover, but first of all, let me talk about insects. It could be that the insects are drawn to the one with the biggest, most flowers, and then while they're there, they go next door. So that you could imagine that effect could occur. To avoid the nutrient thing, we did it the best we could. We used a machete and cut the soil between the plots. So at least there were not roots transporting nutrients across. That doesn't mean that the soil could have been leaching them a little bit. So so cutting the plots was the best we could do. And it turned out, it was good enough to get significant results, even though it probably wasn't perfect.
Is it better to do it like the whole thing is just like one experiment, like not 4 plots?
So how do you mean?
Like if you put salmons in 1 plot and then like go further distance and then like control.
Yeah, that would be the other way to do it. We could have. Yeah. And I probably would have wished we'd done that if we hadn't gotten a significant effect. But we got away with it. It's pretty nice. For practical reasons and all sorts of others, it's pretty nice if you can have this randomized block design. It's kind of a classical thing in ecology, and I think the spilling over fortunately didn't confound us too much. But yes, that could have been another way to do it. I guess the other thing though, Ruthie, is that these salt marshes are more complicated topographically than it looks like from the air or from the photo. So I could take 3 steps here and I might be this much lower, right? And or there could be, well, you would avoid it, but there could be a creek running through it. So, you know, it'd be very easy for there to be localized differences that could make it messier to interpret the results. So having them close together, that was one of the advantages of it.
And controlling for the environment.
Yes, because you know, it's a meter by a meter, so you're only going from here to here, basically, is our square, a little bit more than that. That was a lot better because it is pretty rough walking, believe it or not. You could be heading to if you're not on the trail, you're heading to the pod, and the next minute somebody disappears. Literally, where did they go? And it's because they stepped down into a hole, and there's tall grass, tall sedges, and so it is a little it's complex.
Thank you.
Yes, you bet. That is a question for Young.
Did you ever notice a difference in like different tree composition in comparison to like rivers with more or less sediment?
I don't think we got much of an effect. We did not publish it in that. We published almost everything we had in one paper, and it wasn't in there. And I don't remember what we ended up doing, but I think we didn't get very much of an effect. Other people, in particular Tom Reinke at the University of Victoria, have done work on growth rates of trees by looking at tree rings. It's really technically difficult. It took him a long time to crack the problem, and we used to talk about what he was doing and some of the challenges of interpreting the stable isotopes in the tree rings. There were many other things going on, and so I never touched that. He was working on it, and that was his thing. But he certainly has shown faster growth rates, at least a couple of species of trees that he's nearer to salmon. Basically, he is picking up. He has picked up some salmon effect, and he's published a couple of really nice papers. He and his team on that.
Yeah, I was listening to sound as my first ever ecology professor.
Yes. Yeah. And in fact, he used to work in Alabama, and Morgan Hawking, my postdoc, was his PhD student. So I'm kind of standing on the shoulders of a giant there, and he was no longer working in the territory, but a couple of my sites, he mostly studied 3 watersheds, and I'm studying all 3 of those plus a lot more.
Thank you.
Awesome. One more round of applause.
Okay, thank you.
So much. That was fascinating and awesome. Thank you so much for sharing your time and knowledge. And thank y'all so much for joining us this evening. This happens every Tuesday from now until the end of the summer. If any of y'all are sticking around for another course, maybe not, I don't know. There's Between Up tomorrow, Deep Sea on Thursday back here at 7. And other than that, please enjoy the rest of your evening. Any announcements? Anything else?
No, I think they know. I think you know what you're doing. Yes. No, I think we're good. And I appreciate your moral support for coming out for the story. I'll leave that there.
Thank you so much.
All right, thanks.